On this channel, we have been talking a lot lately about the role of autoimmunity in ME-CFS and the symptom burden that it seems to create, in particular focusing on something called functional autoantibodies. But does this same phenomenon also occur in postural orthostatic tachycardia syndrome, i.e. in POTS? The answer seems to be yes, and in this video we're going to be talking all about that. But first, if you are new to this channel, my name is Patrick Usher, and this is a place where I aim to discuss the research into ME-CFS, Long Covid and related conditions in an accessible way so that you can be more empowered into understanding these conditions better. It's also a place where I talk about the various treatment strategies that I have tried and I'm trying in order to improve my condition. So if that sounds of interest to you, please do like and subscribe. So I have a whole range of recent videos talking about autoimmunity in ME-CFS. And autoimmunity in ME-CFS seems to revolve around something called a functional autoantibody, or rather a group of functional autoantibodies. Now these autoantibodies are against something called G-Protein Coupled Receptors, and they're a family of autoantibodies that, if they exist in the body, are capable of blocking various core physiological processes. For example, they may interfere with the heart's capacity to pump. They may interfere with the perfusion of blood into the muscles or into the brain. They may interfere with the creation of hormones, or they may interfere with the creation of moisture in the body, like sweat and mucus, etc., etc. In fact, they can interfere with a lot of things. You can go and watch my previous videos if you want to learn more about that. So this raises the question, given that ME-CFS is very similar to an illness like POTS, does this same phenomenon occur there? Are there functional autoantibodies against G-protein coupled receptors in POTS? And so we have an answer to this question from a paper from just a few years ago by Grubb. So his paper was called Postural Orthostatic Tachycardia Syndrome is associated with elevated G-protein coupled receptor autoantibodies. And what they did in this paper is they took 55 patients and they measured the level of G-protein receptor autoantibodies in each of them. So this meant that they actually measured these 55 patients for 9 different autoantibodies. They did anti-alpha-1, anti-alpha-2, anti-beta-1, anti-beta-2 and anti-muscarinic 1, 2, 3, 4 and 5. And they did this using the CellTrend testing kit from Germany, a place which is the kind of go-to for people to test for these autoantibodies. It's a test I've done myself. And so as for the results, they were quite interesting. Now, not everyone came back positive for autoantibodies, but there was definitely a trend in which patients did. Now, what was most striking from the results is that over 90% of patients came back positive for alpha-1 receptor autoantibody. As you can see in the graph here, very few people came back positive for the other adrenergic receptor antibodies. Alpha-2, beta-1, beta-2 is a much lower percentage of patients, but a significant, very significant number of patients came back positive for alpha-1. Now, this is quite interesting because the alpha-1 receptors are normally responsible for the return of blood to the heart when you're upright, and they're called the capacitance blood vessels. So normally these receptors will receive a signal when someone stands up to constrict the blood vessels that are central going back up to the heart, and therefore help with an efficient return of blood back up to the heart while someone is standing. If you have autoantibodies against alpha-1 receptors, in contrast, that action will be interfered with, and therefore the blood vessels will not be able to constrict adequately when someone is standing, thereby leading to a reduction of blood flow while orthostatic, which of course then will lead to a compensation where the heart has to beat faster and the tachycardia that comes along with that. So that makes perfect sense in relation to POTS. So this was a very interesting finding. Anyway, as we've just seen, the other adrenergic receptor autoantibodies were positive in a few people, but not in everyone. Now let's look at the muscarinic autoantibodies. So this is very interesting as well. You see that there's a high percentage, just around 50% of patients had high autoantibodies for muscarinic 4 receptors, which also have a role in the heart, and they have a role in dopamine release as well. So we know that, for example, mice who have problems with muscarinic 4 receptors are more likely to be addicted to cocaine. God bless them. Here's a mouse in a laboratory who's looking for the cocaine. And so perhaps if people, if you find that since you develop POTS, problems with willpower or you've become a bit of an addict, hopefully not to cocaine, but maybe to chocolate or whatever, you've less willpower, this may explain that. But anyway, what was really interesting here, we see that, also about 30% of people positive for muscarinic 2, muscarinic 3 receptors. Muscarinic 3 receptors are involved in producing moisture in the body. So if someone is dry, if they have autoantibodies against muscarinic 3 receptors, if they no longer produce a lot of sweat or tears or mucus, etc., that might be explained by that. But what's interesting thing that the study authors found is that, surprisingly, elevations of muscarinic receptor autoantibodies appeared to be dependent upon elevation of autoantibodies against the alpha-1 adrenergic receptor. Exclamation mark. How often do you see an exclamation mark in a scientific paper? Not often. That means this is very important. Now, so what this basically means is that in order for people to have elevated muscarinic receptors, they had to be also testing positive for the alpha-1 receptors. That's very interesting. Now, what does muscarinic mean? Muscarinic means parasympathetic. So the autoantibodies in this case against the muscarinic receptors are against the parasympathetic nervous system. Ultimately, they do lots of other things too, but ultimately that's what's going on. So it's quite interesting that in order to have those, you also needed autoantibodies against the alpha-1 adrenergic receptor, which to me suggests some kind of link with the autonomic dysfunction and the excessive flight or fight state that is ongoing in these conditions. This is just me giving my own idea on this. I don't know how valid it might be, but the idea being that if the autonomic nervous system is in this stress state, it's pumping out a lot of stress chemicals. It may also upregulate autoimmune activity against receptors that are normally associated with the stress response, alpha-1 receptors, and then following on from that, it may elevate autoantibodies against the parasympathetic nervous system. So in this way, the autoimmunity actually mimics the dysautonomia that's happening. And so perhaps once it all becomes established, this autoimmunity, it then feeds right back into the dysautonomia by further clamping down on the parasympathetic nervous system, as well as further contributing to the sympathetic one. So from this study, we have an indication that functional autoantibodies against G-protein coupled receptors are also problematic in illnesses like POTS, not just in ME-CFS. And this makes sense given how much of an overlap there is between these kinds of conditions. Therefore, in conclusion, this paper finds that these results support the growing body of evidence suggesting that POTS may be an autoimmune disorder. It is very interesting that both sympathetic and parasympathetic nervous system receptors are immune-mediated targets. Of particular interest is the apparent tendency that subtypes of both adrenergic and muscarinic receptor antibodies are not detected in the serum of POTS patients unless autoantibodies were expressed against the alpha-1 adrenergic receptor subtype. This phenomenon requires further investigation. So there we have it. This paper does support the idea that there is also something going on with functional autoantibodies against G-protein coupled receptors in POTS and not just in ME-CFS and Long Covid. Now, having said all of that, after the paper came out that we just talked about today, there was a paper subsequently from a different research group that argued the total opposite, that actually said that the results of the study we've looked at today were not particularly valid, and that actually autoantibodies, these functional autoantibodies, are not so important in POTS. They do not seem to be that different if you compare the number of autoantibodies in a POTS patient sample with the number of autoantibodies in a healthy control. There's not enough of a difference to suggest that these functional autoantibodies have any clinical significance. Now, in the next video, we're going to look at that paper, and I'm going to argue against it and actually suggest that these functional autoantibodies are important and that they are a key component of one of many, many components, but a key component in these kinds of illnesses. So if you're interested to learn more about the controversy surrounding these functional autoantibodies, do stay tuned for that video, which will be released in a few days. So that's it. Leave your thoughts down below. What do you think about this research? What are your takes on functional autoantibodies? I look forward to engaging with you in the comments. If you want to learn more about me, my books, I've written one about POTS, I've written one about thirst in ME-CFS and POTS and Long Covid, which is available for free, as well as my consultation service. Please head over to patrickusher.com.